Hi, Victoria. Hi, Kirko. Come up to the stage. Nice to see you.
Hello. Nice to see you both, Katarina, Kirko.
Yo, how you guys doing,
Good,
gals?
good. How are you?
Pretty good, man. About to run all these last minute errands before this goddamn hurricane from Chicago. So we not used to this type of weather. They said it's supposed to be like 7 to 10 inches of rain. I say rain. I'm used to snow, but rain. I don't even know how you gauge that.
Yeah, that's scary. I hope you guys stay safe. So, I don't know. I went through like minor hurricanes, but nothing really bad happened where I live. Like in other parts of Brooklyn, there was like stuff going on, but yeah, flatly I live a little bit up the hill, so usually we're fine. But yeah, I hope everything will be fine. We had really bad blizzards when we lived on Cape Cod on Massachusetts, which is an island, so there I was kind of scared. It was really strong wind and very cold. Oh, hi Orly. How are you? We are talking about hurricanes. How are you today? To unmute, it's all the way on the bottom right hand. There's a little microphone symbol. If you want to speak. Can you hear us, Orly?
Orly, hello. You might find success if you leave and come back.
I can hear you. Hi, everyone.
Great. We'll start in around 6 minutes, so we still have time to hang out. Yeah. Kirko said he's preparing for the hurricane, and I was talking about really bad blizzards I was in on Cape Cod. It was really horrible. But yeah, I hope everyone stays safe.
I jumped in. I just was listening when you said that about the blizzards, and I thought that you were talking about lizards. And so I was really interested to hear what kind of dangerous lizards I'd never heard of that are living on Cape Cod. Um,
Yeah, it was blizzards. So we moved back from Portugal in like the worst year of Massachusetts, having like the horrible blizzards. We couldn't see our backyard, I think, until early May. Like Boston didn't know what to do with the snow, and the ocean froze, even on Martha's Vineyard. I don't know. It was really, there was boats couldn't reach people, so classmates of my son, they lived on these little islands and they couldn't get to school and they were starting to run out of food. It was like very dramatic winter.
that sounds kind of picturesque, though. Were you out photographing?
Back then I didn't do the photography yet, but it was like, there were people playing. So we had the Marine Biological Laboratory has a saltwater pond, so kind of to put their boats and stuff, and the salt water was completely frozen, so people were playing ice hockey, but you also had then frozen ducks in the morning. And but yeah, it was kind of very special. It was like, you know, it's looked like Winter Wonderland somehow, but it was also kind of dramatic because people were out of power for like weeks. Because then the next one came, and the next one, it kind of didn't stop for like 3 months. So anyhow,
So I'm reading 22 Feet of Snow in the room chat. How long does it take to leave a home if you're under 22 feet of snow?
well, we were not allowed outside for almost a week to drive. So we really had to pack up food and stuff because it was not just a lot of snow, it was storm, real storm. And we gladly bought, I never, like, we arrived, it was this horrible winter, so my husband said, okay, let's buy a pickup truck. And then we sold it when we moved away. So that was very good, very fortunate decision because it was the only way to get somewhere and to
I'm
get
thinking,
to,
sorry, go ahead.
yeah, to get also to the institute. I couldn't get away like without that
Well, I was
thing.
thinking because also you needed to load up your truck for all the food that you would need and supplies during the 22 feet of snow that's on you.
Yeah, in the summer, so my son, he was a teenager, so we bought a little boat that he could um, pull with the truck. So friends of him from Portugal came, so they could go around these little islands around Falmouth. So that was fun also. So yeah, if you're on Cape Cod, if you have a boat and horrible winter, a pickup truck. Hopefully they're electric ones soon. So I pre-ordered the Cybertruck, but anyway.
Oh my gosh. I wish it was prettier. The design is so lacking and harsh and unbeautiful.
I
But
really like
congratulations.
it.
I should not, I'm sorry, I apologize. I should not criticize your
Oh
truck.
no,
It's
I
gonna be great.
it's totally fine, but I actually like the design. Like I like the rough, like it looks like cyberpunk type of. I don't know. I kind of like it.
As it should be.
I think we can slowly start. Only thank you for being so patient and waiting here and listening to our conversation. I hope it wasn't too, you know, too annoying. So, welcome everyone to the Science Society. And of course, a special welcome to our guest speaker here today. And before we start, let me give you a short introduction so you get to know our guest speaker a little bit, and then we'll go from there. So, Dr. Orly Lazarov, she's a professor of neuroscience in the Department of Anatomy and Cell Biology at the University of Illinois at Chicago. And she was a graduate student at the Weizmann Institute of Science in Israel. And she was part of the research group of Professor Michael Schwartz. And there she studied the crosstalk between the immune system and the central nervous system in relation to nerve trauma. And she won the Feinberg Graduate School Fellowship of Distinction for her outstanding achievement in studies and research, and the Feinberg Graduate School Award for her distinguished PhD as a distinguished PhD student. And then she joined the group of Professor Sangram Sisodia at the University of Chicago, where Dr. Lazarov studied the multiple aspects of Alzheimer's disease and neuropathology. And she was pioneering studies showing that in addition to genetics, environmental factors play a really major role in the formation of Alzheimer's disease. And then Dr. Lazarov joined the Department of Anatomy and Cell Biology at the University of Illinois in 2005, where she has now her own research group and studies neurogenesis and plasticity in aging and in Alzheimer's disease. And yeah, usually we start with a short interview. So, Victoria, the stage is yours for the interview questions. Thank you.
Thank you so much, Katarina. Orly, that was such an interesting CV to listen to, and I can't wait to hear your talk. And Science Society is so happy to welcome you here. Um, so, to begin, Science Society is all about people exploring knowledge together. So, my questions will allow us to learn a little bit about you aside from your work. And what I would like to ask you is, can you tell us a little bit about a time that you might have noticed that you had an affinity toward science at any time in your life? Maybe when it first showed up for you, maybe as a child in a class, or a relative, shared something with you that you found interesting and you felt like science is my thing. So that's my question.
Well, first of all, thank you. Thank you for inviting me to join this club and give a talk. It's really a great pleasure to be here. And it's hard to say when the whole thing started. I guess during high school, I was very intrigued by you know um, animal behavior, and I wanted to do some field research in Africa. So I decided to study life sciences, and then I realized that I like molecular biology and genetics better than animal physiology, and the rest is just history. So I guess that was probably a turning point.
Thank you. It's to realize what can happen when exposure to topics or aspects of life that we aren't familiar with, and how that opens up our creativity so we can find our path. And so, speaking of path, can you tell us, maybe give a brief overview of your journey from that point when you became more interested in molecular bio and genetics to your work that you're about to share with us today?
Yeah, I think it was a real evolution of interests. Um, I guess I think it is really interesting because science is all about questioning, right? It's all about not taking things for granted, but always questioning the dogma. And I guess it was just an evolution of studies that I've read and was very intrigued by. The brain, I think, has always been a major interest of mine, and how to develop therapy for brain disorders has always been a major challenge. And I thought that this is something that I would like to learn more about. And during my graduate years, I was fascinated by different aspects of brain plasticity and regeneration. And when I started um, learning more and more about Alzheimer's disease, I realized that, and I will talk about it a little bit. I realized that there are aspects that are unexplored in the field, and I was very intrigued by that. And one of them is hippocampal neurogenesis that I'm going to talk about today. Um, so about 20 years ago, when I started researching, I realized that there's very little that is known, but actually I could see the connection between hippocampal neurogenesis and Alzheimer's disease very much early on, and the rest is history. I'll definitely talk about how that evolved into the work that we are presenting today.
So fascinating. Thank you. I have one more question, if I may. How, if you know, how is it that you felt comfortable questioning the dogma, as you mentioned, when other people may not feel so comfortable? What do you think it was that gave you that courage to trust yourself and your own questioning?
I do believe that we have to live our lives questioning everything every day, right? So um, it's just, I guess, the acknowledgement that research tools are, as wonderful as they are, they're also limited. And if we acknowledge the limitation of our own tools and realize that we can show only what our tools allow us to show, then you come to the realization that as tools develop and we have more opportunities to reevaluate previous data, we should do that. And I guess it's not me being you know braver than others. It's just you know something that I guess I've been trying to implement in my personal life, and it you know obviously translated into my research as well.
Thank you. I appreciate your answers. They're fascinating because also, aside from your research, we never know what sort of example or lesson we'll pick up from hearing someone's journey. And so it's always important to share and learn. So at this point, you're welcome to go into your talk, and if you'd like to have the Q&A following your discussion, then that's great. And if you prefer to have questions from the audience and people on stage to drive the shape of your discussion, then that's fine too. We, the moderators, are here to help make that happen. And sometimes people will put questions that they have for you in the room chat, and we can let you know about that as well. So all you really need to be concerned with is delivering your talk and enjoying the time here. So did you have any other questions beyond that?
No, that's great. Thank you so much. I am happy to take any questions during the talk. I just need someone to speak up because I may not realize that there is a question somewhere. So if anyone has a question, please speak up. Otherwise, I'm happy to take any questions at the end of the talk as well. So whatever works for you guys.
Fantastic. Okay, we will moderate the questions and we'll be sure to let you know. And Katarina has got your PDF right there. So the mic is yours. Thank you so much, Orly.
Okay, thank you. All right, so good afternoon, everyone, or almost good evening. Depends on where you are in the world, I guess. And thank you again for the invitation to speak today. I would like to talk about our research in the last several years. We had very interesting observations concerning the role of hippocampal neurogenesis in Alzheimer's disease. So let's start talking about Alzheimer's disease first. We'll go to the next slide. And I cannot see the slide, so I'm not sure what slide you're pointing at, but if we assume that the first slide with my name on it is the first slide, then the next slide, the second slide, will show some information about Alzheimer's disease. So we're talking about a neurodegenerative disorder that is characterized by progressive memory loss and cognitive decline eventually leading to dementia, which is the inability to perform daily life independently. In some cases, we also see altered behavior such as paranoia and other behavioral abnormalities. The interesting phenomenon of neuronal vulnerability is a very important part of Alzheimer's pathology. An area that is called the hippocampal formation, which includes the adrenal cortex and the hippocampus, exhibits high vulnerability of neurons. And in fact, neurons in the adrenal cortex die first in the disease. In about mild form of Alzheimer's disease, 60% of layer 2 of the adrenal cortex die. And in a more advanced stage, almost 90% of this layer is lost. And I'll talk a lot about the hippocampal formation. This is an area that is extremely important for memory formation, and we'll discuss that extensively today. In addition to neuronal vulnerability, the two pathological hallmarks of Alzheimer's disease are amyloid deposition, which are A beta aggregates that are a cleavage product of amyloid precursor protein, and neurofibrillary tangles, which are intracellular aggregates of hyperphosphorylated tau. There are other pathological hallmarks such as inflammation and gliosis and others. As you can see in the picture, you can see Dr. Alzheimer, who is the psychiatrist who discovered Alzheimer's disease. And next to his picture is the picture of his patient, Augusta D., who had a genetic form of Alzheimer's disease. So what is the difference between genetic and non-genetic Alzheimer's disease? The genetic is a familial form. It is an autosomal dominant form that is caused by mutations in either amyloid precursor protein or APP, presenilin 1 and presenilin 2. However, the vast majority of Alzheimer's cases do not carry these mutations, and they're caused by probably an unknown, would be fair to say, factor or factors. There are risk factors for the sporadic late onset form of Alzheimer's disease, such as age, ApoE4, aging, of course, TREM2, and other factors that you may be familiar with, such as type 2 diabetes, vascular disease, cardiovascular disease, are all risk factors for this disorder. So what do we know about Alzheimer's disease so far? We know that it's a very slow progressing disorder that we presume starts decades before the clinical diagnosis, so before memory loss becomes apparent. We know that aging is the greatest risk factor for Alzheimer's disease. So hypothetically, if we're able to prevent aging or delay aging, it would reduce the number of cases of Alzheimer's disease. We know that so far the association between level of pathological hallmarks, namely neurofibrillary tangles and amyloid deposition, and the cognitive diagnosis, which means level of impairments of memory loss, is not entirely clear. We do know that there are people with extraordinary memory called superagers that in their brains we could find neurofibrillary tangles and amyloid plaques. And so this association between the pathological hallmarks and Alzheimer's disease diagnosis is not fully understood. It is definitely possible, and I would think that some of the people in the field believe that what we are facing today is actually the realization that Alzheimer's disease is in fact an umbrella of subphenotypes and pathways. Different individuals could have different risk factors that would trigger molecular pathways that may be different, but would lead to the same end result, which is memory loss. But ultimately, if we're thinking about Alzheimer's disease, to date, the cause of memory deficits is not fully understood. We don't really fully understand the mechanism that causes the dysfunction of memory circuits in Alzheimer's disease. So the memory circuit, as I mentioned before, is composed of the vulnerable neurons in Alzheimer's disease. We're looking here, and this is, we're in slide 4 right now. I'm sorry forgot to mention that I'm moving a slide. So this is slide 4 right now. We're looking at the hippocampal formation, and you can see that neurons in layer 2 of the entorhinal cortex project into the dentate gyrus of the hippocampus. An interesting phenomenon and very unique of the dentate gyrus of the hippocampus is the existence of the neurogenic niche, which is a niche of neural stem cells that exists in the hippocampus, in the dentate gyrus specifically, throughout life. And from neural stem cells, which are multipotent, new neurons are being generated and incorporate in the granular cell layer of the dentate gyrus. They become granular neurons, and as you can imagine, since layer 2 of the entorhinal cortex projects into the dentate gyrus, new neurons play an equal role in the granular cell layer as those more mature neurons in terms of future role in the dentate gyrus. Now, this is a very unique phenomenon because in other areas or the vast majority of the brain, there are no stem cells. So neural stem cells is a very unique phenomenon that is actually in the rodent brain. We find in the hippocampus, in the dentate gyrus, in the granular cell layer of the dentate gyrus, and in the olfactory bulb, or in the subventricular zone that is connected to the olfactory bulb in that sense, because neural stem cells that differentiate into mature neurons migrate into the olfactory bulb. So indeed, only so far, 2 areas that are confirmed to have neural stem cells that differentiate into mature neurons, and the hippocampus is one of them. Now, the big question is, what is the role of new neurons in the dentate gyrus, and why in the dentate gyrus specifically, an area that is so important for learning and memory? So here in the next slide, you can appreciate that these are neural stem cells that have the capability to differentiate into all 3 types of brain cells, either oligodendrocytes and astrocytes, or into mature neurons. This process of neurogenesis takes about 4 to 6 weeks. During this process, neurons, or the new neurons, exit the cell cycle as progenitors and mature into new neurons. They're expressing different neurogenic and neuronal markers, and they are thought to play a major role in hippocampal function, such as pattern separation, associative memory, spatial navigation, and so forth. So to summarize this, we have here a phenomenon where new neurons are being incorporated in the dentate gyrus of the hippocampus throughout life and play a role in hippocampus-dependent learning and memory. Now, a big question is, does hippocampal neurogenesis exist in the human brain? Because if we are thinking about translating any potential findings into a therapy of the human disease, the human brain, we would be very interested to know, and I'm moving to the next slide, whether hippocampal neurogenesis does exist in the human brain. And indeed, several studies have shown from Fred Gage's lab and from others that indeed in the human brain, neural progenitor cells become mature neurons in the dentate gyrus. And very intriguing studies from 2013 have shown that approximately 1/3 of the neurons in the dentate gyrus are turnover, and that indeed every day in the human brain, about 700 new neurons are being added to the hippocampus. So I would encourage you to multiply 700 new neurons times 365 days times 80 years, and you will get a lot of neurons, a lot of new neurons that are being added to the dentate gyrus throughout life. However, this has been really questioned very seriously, but by several groups, and who reported that they could have not found any new neurons in the aging human brain. And so, to examine this possibility and to really assess for ourselves whether hippocampal neurogenesis does exist in the human brain or not, and what happens in Alzheimer's disease, and I'm moving to the next slide, we obtained postmortem brain sections from the Alzheimer's Disease and Research Center at Rush University. And those postmortem brain sections were of three major groups. One group was normal aging, so individuals that had intact memory when they passed away. The other group was of individuals that, when they passed away, had a diagnosis of mild cognitive impairments, which is the precursor, could be a precursor to Alzheimer's disease. And then the third group was of patients that, when they passed away, they had a clinical diagnosis of Alzheimer's disease. And we examined the brains, and I'm moving to the next slide, and asked whether, first of all, whether we're able to detect new neurons in the human brain, and second, whether we're able to find any differences between normal aging and Alzheimer's disease. And indeed, as you can see here in the graph on the upper left, we observed that in the normal aging group, which is designated here as NCI, no cognitive impairments, we observed, and you can look at the pictures underneath, and you can see that this confocal imaging of new neurons, DCX is double cordon, in short for double cordon, which is a proxy of new neurons. We observed that in the normal aging, there are there's definitely a substantial amount of new neurons that are being generated in the dentate gyrus of the hippocampus. Interestingly, we observed that in NCI and AD brains, the number of new neurons was significantly lower. We further looked to see whether we're able to detect any pathology in those new neurons specifically. And indeed, it was really interesting to find that, and this is in the next slide, you can see that in double cordon positive, DCX positive new neurons, in the Alzheimer's brain, we could detect more hyperphosphorylated tau. And this is something that I think is pretty novel, because I don't think that Anyone has described that before. And I think, many people in the field would probably argue that impairments in neurogenesis may be a result and not a cause. And this is, one of the earlier times that, you know, that would be an appropriate time to say, well, maybe not. Maybe this is something to rethink. Um, So interestingly enough, when we looked at the amount of new neurons as a function of cognitive diagnosis, we indeed realized that having more new neurons reduced the likelihood of cognitive deficit diagnosis, suggesting that, and this is in the next slide, suggesting that the more new neurons one has, the likelihood of having a clinical diagnosis of Alzheimer's disease is reduced. So, in other words, the number of new neurons in one's brain correlates with the level of diagnosis of a positive diagnosis. So that was very exciting. And this is all in the human brain. And so we went back to understand the mechanism underlying neurogenesis in Alzheimer's disease and understand whether indeed impairments in hippocampal neurogenesis in Alzheimer's disease are part of the mechanism of memory failure. So in the next slide, you can see that I outlined here some milestones of research in mouse models of Alzheimer's disease. And here, we're getting back to the mouse models because, our ability to look at molecular mechanisms in the human brain is more limited because we're looking at an endpoint. So in order to understand mechanisms, we're going to get back to our research in mouse models. And I'm going to show you how our research evolved during the years. One of the first observations that we've made was that hippocampal neurogenesis is impaired in mouse models of familial Alzheimer's disease. And if you go to the next slide, you will see that when we look at brain sections of mice that express mutant forms of familial Alzheimer's disease, and on the slide, this is slide number 13, I indicated several mouse models that we and others have been looking into. You'll see that the number of new neurons is significantly reduced in mouse models of Alzheimer's disease compared to wild-type mice. And this takes place very early on. For example, in some mouse models, it could take place at 2 to 3 months of age in mice that have a very extensive pathology, and in mice that have slower pathology progression, this could take place around 5-6 months of age. So very, very early in the pathological progression of Alzheimer's disease, which really suggested to us that this could be part of the molecular mechanism of memory deficits rather than a result of the pathology. So we kept looking into the mechanism, and one of the most intriguing observations that we've had, I think, was that those proteins that cause familial Alzheimer's disease are actually regulating hippocampal neurogenesis. And I'm looking at slide 15 now. And here I indicated some characteristics and some observations that people have made during the years, including ourselves, about the role of presenilin 1 and amyloid precursor protein as regulators of hippocampal neurogenesis. So, as some of you may know, presenilin 1 is thought to be the catalytic core of gamma secretase that cleaves several or numerous, I should say, substrates. Some of them play a role in neurogenesis, such as Notch1, ERB4, APP, L1, etc. In addition to that, an observation that has been made long time ago, and I guess I wouldn't say went unnoticed, but I would say that was probably partially overlooked, is that the ventricular zone was substantially thinner in presenilin 1 knockout embryonic um, embryonic mice, and the number of neural progenitor cells was dramatically reduced. Later on, we have shown that even during adulthood, presenilin 1 regulates the differentiation of hippocampal neural progenitor cells. And likewise, and I don't have time today to talk about all those studies, but if those of you who would be interested, I indicated our studies on the slides, and you're welcome to look it up. But one of the examples that I decided to include today is that when we downregulated the level of presenilin 1 in neural progenitor cells, we observed that neural progenitor cells exit the cell cycle. You can see here on slide 16, you can see that the number of proliferating cells is reduced in brain sections where we injected lentiviral vectors expressing presenilin 1 siRNA. And the number of new neurons, or the number of newly mature neurons, is upregulated. As you can see here, neurons that are GFP positive, BRDU positive, and beta 3 tubulin, these are new neurons, and their number is increased. And the reason is because presenilin 1 is a regulator of neural progenitor cell differentiation. So what do we learn from that? We learn from it that indeed presenilin 1, which causes mutations in presenilin 1, cause um, familial Alzheimer's disease, is in fact a regulator of hippocampal neurogenesis, which is quite remarkable. I don't think, I don't think that we anticipated that 20 or 30 years ago. And indeed, to elaborate on that point, when we analyzed the morphology of new neurons that had a reduced expression of presenilin 1, we observed that their dendritic spines and the number of dendritic branching were all compromised. And this, you can see that on slide 17. And their survival, also on slide 18, you can see that the survival of new neurons and mature neurons that had reduced expression of presenilin 1, which is, just to clarify, reduced expression of presenilin 1, we think that mimics the real function of presenilin 1 mutant forms, because we already know that gamma secretase activity is reduced in the mutant forms of presenilin 1. And indeed, when presenilin 1 is reduced, we can see that there are less new neurons that are being that survive in the granular cell layer of the dentate gyrus, and their survival is compromised, which means the dentate gyrus layer is potentially thinner or less functional, which is I'm yet to show you this data. When we looked at the molecular mechanism of a reduction of presenilin 1 expression in neural progenitor cells, you can see here, and I'm on slide 19, you can see that levels of markers or proxies of stemness, such as nestin, and proxies of proliferation, such as cyclin D1, EGF receptor, PDGF receptor, are downregulated, while proxies of differentiation, such as neurofilament, are upregulated, suggesting that downregulating presenilin 1 in neural progenitor cells indeed enhances their differentiation, but also compromises their survival, their morphology, and their function. And when it comes to their function, you could see in, again, slide 19, you could see that levels of phospho-Creb, for example, a major regulator of neural progenitor cell survival, is downregulated. In addition, phosphobeta-catenin, very critical for their function, is also downregulated in those new neurons. So then the question was, okay, if we know that hippocampal neurogenesis is impaired in familial Alzheimer's disease, and we also partially know why, and shown that APP and presenilin regulate these processes, then the question is, if we deplete hippocampal neurogenesis, would that exacerbate learning and memory in Alzheimer's disease? And that was a pretty difficult task because Alzheimer's mice have impairments in learning and memory quite early on. But indeed, and I'm on slide 21, and this is a task that is relatively easy. It's a task that mice have to discriminate between two different contexts. And in the first context, they get a shock. In the other context that is very different, they don't get a shock, but they need to remember that they got a shock somewhere else. So they need to be able to discriminate between the two different contexts. As you can see here on slide 21, wild-type mice are easily performing this task. They learn it from day 1, and they're able to perform it through day 3 very successfully. When it comes to the Alzheimer's mice, you can see that Alzheimer's mice do not perform well on the first day, but then they'll learn the task and perform it successfully on day 2 and day 3. When it comes to mice, the familial Alzheimer's mice with reduced neurogenesis, you can see that they fail to perform the task on day 1, 2, and 3. So they were not able to learn the task at all. That strongly suggested that neurogenesis plays a major role in memory deficits in Alzheimer's disease. So then we asked the question, well, if this is the case, if we rescue neurogenesis, are we able to rescue memory? So in order to do that, we let the mice experience environmental enrichment, which is a setting that where mice can run. And I can show you, actually, I can jump into slide 26, so you can see the large cage with the toys and running wheels and a lot of stimulation for the mice, and they're able to run on the running wheels and so forth. And indeed, we observed that those mice that experienced environmental enrichment exhibited greater level of neurogenesis. You can look in slide 23. You can see that mice that experienced environmental enrichment had more new neurons that incorporated in the granular cell layer of the dentate gyrus. And again, those of you who are interested in more details, the papers that we published are indicated on the slides. So the issue with environmental enrichment is that this is a stimulation that affects numerous aspects of the hippocampus, not only hippocampal neurogenesis. And so the question was, if we are specifically augmenting neurogenesis, are we able to restore memory formation? And that was the main next thing for us. So on slide 25, you can see our hypothesis. Augmenting neurogenesis may ameliorate memory deficits in Alzheimer's disease, and new neurons play a role in memory circuit in Alzheimer's disease. So indeed, instead of environmental enrichment, we chose a genetic form of augmenting neurogenesis, and I'm on slide 27 right now. And here is our experimental model, and this is we used animals that were given to us from Dr. Renee Hen from Columbia University. And on the left, on the upper left, you can see that what we have done was to breed into our familial Alzheimer's disease 5-FAD mouse model. We've bred two mouse models, one that expresses the Cre recombinase under the nestin promoter, and the other one that had a floxed Bax gene. And what this combination does is that it enables us, with the administration of tamoxifen, to excise or to delete Bax, which is a major regulator of neurogenesis. And when Bax, which is an apoptotic gene, is deleted, we're able to increase the level of neural progenitor cells that survive. And as a result, the result is that we get more new neurons that are being generated and incorporate in the granular cellular. So on the right side here, you can see the quantitative analysis. And indeed, when we injected tamoxifen into those 5-FAD mice, the Alzheimer's mice, we observed increased level of neural progenitor cells and new neurons that incorporated in the granular cellular of the dentate gyrus. So the next step for us was to ask whether we're able to rescue memory when we have increased neurogenesis, level of neurogenesis. So here on slide 28, you can see that we tested those mice with augmented neurogenesis in two tasks that are hippocampus specific. One is novel object location here on the left, and I'm on slide 28. And on the right, contextual fair conditioning. And not surprisingly, the familial Alzheimer's disease mice performed pretty poorly on both tasks. And you can see that compared to the wild type mice in blue, the Alzheimer's mice in red performed very poorly and failed the tests. And those mice with augmented neurogenesis, which are labeled as TNBF, tamoxifen-induced nestin Bax familial Alzheimer's disease, performed almost as good as the wild type mice, suggesting that augmenting neurogenesis in familial Alzheimer's disease restores memory formation in hippocampus-dependent memory forms. Both the novel object location and the contextual fair conditioning have been rescued. So to understand the mechanism by which new neurons rescue memory formation in Alzheimer's disease, we looked to see whether they are part of the memory circuitry. And we have done that by using a construct or an engram kit. Now for that, I have to explain what an engram is. The engram is a set of neurons that participate in the memory process. They get activated either usually during memory acquisition. These are scarce number of neurons in a specific layer or in several brain areas that get activated following memory acquisition, and they participate in memory storage and in memory formation and acquisition. So in order to understand whether new neurons are part of the engram, part of the memory storing circuitry, we used a memory kit that we obtained from Dr. Chenegawa's lab. And you can see it here on slide 29. This is a kit that, it's a viral kit that is composed of a promoter, CFOS promoter. And CFOS is an immediate early gene that designates the activation of neurons. and it is based on the TET-off system, which, in other words, when there's tetracycline is present, the tetracycline prevents the transcription of any gene that is downstream of the tetracycline transcription activator. When tetracycline is not present, um, the the cells that express this viral vectors will appear as green, as you can see here on the right. These are green neurons that got activated and expressed successfully whatever genes are expressed downstream of the tetracycline element, responding element. In our case, it's YFP, which is the green fluorescent protein, as you can see here in the pictures on the right. So using this engram kit, we asked whether new neurons are part of the engram during memory formation. And indeed, what we observed is that the total number of neurons that participated in engram was. They knew that everyone. I'm sorry. Is there any question? Okay. We observed that the total number of neurons that participated in the engram, as you can see here, and I'm on slide 30, is comparable between the three groups, which are the wild-type mice, the mice with Alzheimer's disease, and the mice with Alzheimer's disease and augmented neurogenesis. When we looked at the number of new neurons that participated in the engram, we noticed that the number of new neurons in the engram is significantly and severely reduced in the Alzheimer's mice. And that when we augment neurogenesis, we upregulate the number of new neurons that are being recruited into the engram. Even when we look at the percentage of new neurons as part of the engram, we see that it's compromised in Alzheimer's disease. And following augmentation of neurogenesis, we rescue the number of new neurons that are being recruited into the engram. To understand whether those new neurons were also reactivated during memory retrieval, we looked on the next day of contextual fair conditioning and asked whether these new neurons would be reactivated following memory retrieval. And in order to do that, we looked at another immediate early gene that was activated in those new neurons called EGER1. And indeed, as you can see here, and I'm on slide 31, the majority of new neurons was the number of new neurons that participate in memory acquisition and retrieval was significantly lower in Alzheimer's disease and was rescued in Alzheimer's disease with augmented neurogenesis. And here, these three pie representations of the engram show that in wild-type mice and in mice with augmented neurogenesis, the number of the total size of the engram is comparable, and the number of new neurons that participate in the engram is greater compared to familial Alzheimer's disease, where the number of new neurons that participate in the engram is severely impaired. To determine whether these new neurons that participated in the engram indeed have characteristics that can suggest that they participated in learning and memory processes, we looked at their dendritic spines, and indeed we observed here that those new neurons that participated in the engram following augmentation of neurogenesis had rescued synaptic density in familial Alzheimer's disease, suggesting that in familial Alzheimer's disease, the synaptic density of new neurons that participate in the engram and also of mature neurons on the right side, and I'm on slide 32, is significantly impaired. And when we augmented neurogenesis, the level of dendritic spines in new neurons as well as in mature neurons was significantly improved in all aspects. However, this is so far I've shown you a lot of correlation between the number of new neurons and the performance level in hippocampus-dependent learning and memory, as well as the number of new neurons that participated in the engram. Now, the question is, are these new neurons really required for formation of contextual fair conditioning in familial Alzheimer's disease? In order to prove to ourselves that indeed the augmentation of neurogenesis is what is really required for the rescue of memory, we used DREAD. And for those of you who do not know what DREADs are designer receptors exclusively activated by designer drugs. And these are, we used, in this case, we used retroviral vectors, as you can see here, and I'm on slide 33. You can see retroviral vectors expressing HM4 receptors. When we treated them with the specific actuator with CNO, we were able to inactivate specifically the activity of new neurons during the memory task. And once we did that, we noticed that the rescue of memory was reversed. In other words, mice failed to perform the task when new neurons were inactivated during memory acquisition, suggesting that indeed those new neurons that were augmented in familial Alzheimer's disease were required for successful formation of contextual fair conditioning. Very excited by these observations, I would like to tell you what that means. And here I have a scheme here, and this is on slide 34. This is a scheme of the dentate gyrus, and in green are neurons that get activated and get recruited into the engram following memory acquisition, while in blue are new neurons. In a normal animal, some of the granular neurons in the layer are being activated and are becoming green, including some of the new neurons. However, in Alzheimer's disease, there are less new neurons in the layer, and therefore the engram is impaired and memory is affected and is impaired as well. So what is the mechanism? Why, when we augment neurogenesis and increase the number of new neurons that participate in the engram, we are able to rescue learning and memory? In order to do that, we performed in situ sequencing of mature and immature engram neurons. And this is a very large story that I could have probably spent another hour telling you about, but I'm just going to tell you about it very briefly. And those of you, again, who would be very interested are welcome to read our paper that was just published at the Journal of Experimental Medicine a few weeks ago. And what we observed was extremely interesting. So the way we've done it is we examined whether augmentation of neurogenesis affects the profile of granule cells in the dentate gyrus. So in order to do that, we examined the transcription signature of both immature and mature neurons that were recruited into the engram in the dentate gyrus of the three groups of mice that we had. And we looked at 159 genes of interest that were sequenced simultaneously with single cell resolution during the in situ sequencing. And here is what we observed in very, very briefly, because the time flies, and I think that this, again, could be a very long story. But just to keep a very long story short, what you could see here on the left, and I'm on slide 37, you can see that this is the profile of all neurons that participated in the engram. And in the middle is the profile of mature neurons versus on the right is the profile of new neurons. And just try to just look at the colors, and you will see that on the right is the familial Alzheimer's disease mice with augmented neurogenesis. In the middle is the I'm sorry. On the left is the familial Alzheimer's disease. In the middle is Alzheimer's disease with augmented neurogenesis, and on the right is the profile of wild type neurons. And you can see that there is much greater resemblance between familial Alzheimer's disease, augmented neurogenesis, and wild type neurons. So, in other words, both immature neurons and new neurons exhibited rescued profile uh, following augmentation of neurogenesis in familial Alzheimer's disease mouse models. And again, I'm going to keep this very, very short because I could have talked about it for at least several more hours. But those of you who are interested in the details of this in situ analysis are welcome to look in our papers. So, since I'm running out of time, I would like to summarize the talk and summarize what I've been telling you today, which is, first of all, hippocampal neurogenesis persists in the human hippocampus in aging and in Alzheimer's disease. Second, neural stem cells, progenitors, and immature neurons are detectable in the aging and Alzheimer's brain by protein proxies and their transcripts. The number of new neurons and neural progenitor cells is reduced in Alzheimer's disease compared to age-matched individuals with no cognitive impairments. The number of new neurons correlates with cognitive diagnosis, and having more new neurons increases the likelihood of normal cognitive diagnosis. We notice that deficits in hippocampal neurogenesis plays a role in memory impairments in Alzheimer's disease, that new neurons get recruited into the engram during memory formation, and indeed, defective hippocampal neurogenesis results in an impaired engram that is manifested by memory deficits. As soon as we enhance neurogenesis, we're able to rescue the engram, leading to the restoration of memory in Alzheimer's disease. And this is just to take the time to thank the wonderful students and lab members and collaborators that participated. The human brains were examined by an MD PhD student, former student of mine, Matt Tobin, and by a graduate student at my lab, Amit Dasuki. And they are, and I'm on slide 39. The engram study was performed by Rachna Mishra, a former postdoc, and by Tronga Fan, Muskan Gupta, Pavan Kumar, and Zach Morrissey at my lab. And the collaborators, Renee Hen for giving us the mice, Tanagawa for giving us the engram viral cocktail, David Bennett and his team for the postmortem brains, and Bill Hunter for the analysis of protein interaction with synaptic markers. Bob Vassar is the one who generated the 5 FAD mice. And Mark here on the right, Mark Klein helped us with the analysis of the bioinformatics and in situ sequencing. And thank you so much, and I'm happy to take any questions.
Thank you so much, Orly, for this wonderful presentation and to give us such a great overview of the amazing research you've been doing over the years. It's I agree, we could probably spend many hours talking about the work you did, and because it's also very interesting. So thank you for summarizing it in this way. And it's so impressive, the work that you have done and all the techniques that you've been using. So yeah, thank you again. It's a great honor to having you here. And please, everyone, flash your microphones or raise your hand and to ask questions. And yeah, or use the chat if you would like to use the chat. And yeah, and then we'll go from there. And yeah, I would just like to ask and comment. I really think that it was interesting also in your CV that you were very early on interested in the connection between the immune system and the neural system. I think it was very early that you did this. I know a lot of people are interested in it now, but it was kind of early on, and that was already so impressive to me to read that. And you really showed really well the mechanism that you could even show the integration of the new neurons in the system in a functional way. That is really impressive. And do you think that since you showed this mechanism, how this works, do you think we can translate that also to maybe Parkinson if we would use stem cells, to transplant stem cells into the brain, that by the research you've done, that you could basically figure out how to functionally use the stem cells to maybe also help people with Parkinson and other disorders. Thank you.
Yes, so I think for the research that we do concentrates on learning and memory. And I think that for forms of Parkinson's where there's dementia involved and there's loss of memory, I think that could definitely apply. When it comes to other brain functions that are motor function and so forth, then we're talking about a, a different sort of intervention. We're talking about whether we are able to direct stem cells into other brain areas and have them incorporate in a functional manner and perform the tasks that local neurons perform. And this is a little different, and probably we're probably hoping that one day that will happen. But this will definitely require further research.
Yeah, thank you so much. So yeah, I hope you get all the funding to figure out all of our problems, but not all. But yeah, it's a really amazing. And please flash your microphones, Dr. Shah. And please go ahead, and then Dennis, Kyoko, and Marcus. Thank you so much.
Yeah, thank you for sharing your wonderful work with us. And a couple of questions came in my mind. So we know that about the insulin resistant because you just talked about the tamoxifen. In the same time, at the beginning, I was thinking about the mechanism and kind of glucose metabolism because we are talking about the hippocampus. And I was just wondering during your research, did you, gather any information about the any specific law of the hippocampus and how it's working in a case of the glucose metabolism? This is one part I wanted to ask, and later I will ask my question about the tamoxifen.
So it's interesting that you're asking because we have other studies that I've not shown here today that look into the molecular mechanism that underlies type 2 diabetes induced Alzheimer's disease, which we know that type 2 diabetes is a risk factor for Alzheimer's disease, in that individuals who suffer from type 2 diabetes have a greater risk of deteriorating into dementia. And with that knowledge, I mean, we're still, yet to figure out the mechanism. And there is a whole line of investigations in my lab looking into this. And I think that if you look at my current and hopefully near future papers, you'll probably be able to get an idea of our findings in that regard, and the impairments not only in the dentate gyrus but in the brain, the whole brain, impairments that potentiate the or start the escalation from type 2 diabetes into cognitive deficits and Alzheimer's disease.
Exactly, and when we are talking about the neurogenesis, because as you know that some of the patient, for example, in the children, we have a certain cancer, and if it's appear in the brain, even during the chemotherapy later, they can experience the early sign of the Alzheimer. That's why it's very important from that perspective. Also, I was just wondering, is there any specific interleukin or signaling pathway that we just recognize during your research? For example, specifically, we are talking about TNF or IL-6 or any kind of STAT signaling or such a thing.
I think what you're asking is very important, but I think that it would require us to look more specifically in a cell type specific manner in order to determine whether alterations in interleukins, one or other, take place, particularly because we know that microglial cells are part of the niche, the neurogenic niche in the hippocampus, and there's a very good chance that indeed alterations in interleukin or other inflammatory um, signals are being altered in Alzheimer's disease, and there's definitely a good chance that these are molecular pathways that we haven't elucidated yet, and there's definitely room for investigation.
Thank you.
Yeah, thank you so much. Dennis, please go ahead.
Thank you, Katerina. Thank you, Doctor. This was a really interesting presentation. I had a couple of questions. First one is, were you tracking the behavior in regards to sleep? Was that a factor that you were examining in your study?
It's a very good question. Unfortunately, we have not done that. Sleep has been increasingly associated with Alzheimer's disease, and I think that this question is a very good one. We haven't looked at the circadian cycle of the mice. I can say that as far as our study is concerned, the mice have been on a constant cycle of light-dark cycle, but we have not looked into behavioral sleep patterns in our study.
Okay. And was it just a standard lab diet for these animals, or was there any sort of variability in that metric?
The diet was standard. However, when we used the engram cocktail, the viral engram cocktail, as I mentioned, we had to supplement the diet with doxycycline in order to modulate the expression of the engram cocktail, the CFOS-induced EYFP expression. So this is one aspect, and then Of course, the tamoxifen injection, which is not part of the diet, it's injected IP, is also another factor that we had to take into account.
And then for it, this would obviously be difficult to measure in mice, perhaps. But have you noticed any sort of light sensitivity or inability to look at computer screens in Alzheimer's patients, or in animal models, rather?
Yes. So, the mice have been tested for elevated levels of anxiety, and we have not detected any abnormalities in that regard. When it comes to screen, this is something that has to be done in humans, and I cannot comment on this because we haven't done that. But in mice, it's a little bit more challenging to test.
I can imagine. And then the last question is, and this is more for, again, human studies, but I have seen claims that consumption of certain types of fungus, such as lion's mane, has the ability to promote neurogenesis. I'm not sure of which region specifically, but do you have an opinion or any information on that?
No, I haven't. I haven't heard about that. That's interesting.
Okay, great. Thank you so much for your answers.
My pleasure.
Yeah, Marcos or Kiko, did you have a question?
I don't. This is a great presentation, though, man.
Thank you.
Thank you. Marcos, did you?
Yes, hello. Yeah, so I'm a radiologist, but I used to work in neurology and neurosurgery and psychiatry, but only for like shorter period of times. But and I have a sister who has Alzheimer actually in Germany. What I wanted to mention is, and I'm sure Oli was very nice to present that, is that I'm sure you know that person Dale Bredesen. Is that a guy? It's a neurologist from UCLA who wrote that book, The End of Alzheimer, whatever that means. Have you heard of him? Just curious. Orly,
No, no, I have not.
you have not. Okay, you see, that's the reason I bring it up. Um, it's a very fascinating story. I mean, he's a serious researcher. He's actually more a researcher than a neurologist, quite frankly. But then he's married to a general physician like Dr. Shah, like an integrative medicine wife, and he did a lot of work trying to figure out. He does like a more holistic approach, but he has a lot of data. And some of his data, to me, was when I first heard it, is quite shocking, which vibrates with your theme, that he has published like 10, 20 people with a certain subset of reason to have Alzheimer, that the hippocampus actually, came back, the volume. He measured that on MRI scans, etc. So it's fascinating work. There are a lot of YouTubes about him. And he also talks a lot about, which goes back to Dr. Shah's comment, he basically divides Alzheimer in different subgroups: the inflammatory, the toxic variant, the genetic variant, and glucose metabolism plays a big role. So interval fasting he promotes a lot, and he talks about getting, and I stop in a second. he talks about getting, like, being tested, which I did because of my sister when I'm in the 50s, and see what your predisposition is genetically, metabolically, etc., and then trying to find means to prevent it or do something about it. Fascinating work. I wrote it in the chat, so I think it's worthwhile to look that up.
Well, I think, generally speaking, I think the idea of testing, especially if one has a familial form in the family, it's worth testing. And even if it's not a familial form, right? Even if there's a greater risk of cardiovascular disease in the family, or some kind of cerebrovascular stroke in the family, it's worth testing. Diabetes. And I would agree that there's a lot that we can do. Now, I wouldn't dare to say that we can totally prevent the disease. I would not dare to say that we can at this point cure it. But I would say that there are measures that could potentially um, delay maybe the onset and maybe the severity of the disease. And again, those measures are, almost common sense, right? Which is a diet and exercise and keeping your brain occupied by challenging your brain, right? So there's the term of use it or lose it, right? That is we're all familiar with. Challenge your brain, learn new languages. Now again, this is always a good good methodology for great aging, right? Um, Again, I wouldn't dare to say that this is something that could y- you know could solve the problem, not even close to that, but may help in all aspects as we age and as we become more susceptible to different disorders, including memory loss. So I would, generally think that, it never hurts to live a healthy lifestyle. And that could prevent not only dementia, but a whole array of brain and systemic disorders. Um, that said, I do believe that we will have to have some sort of intervention in order to fully prevent and cure pathology.
If I may, really quick follow up. That is maybe a little bit related. We had here a few months ago Dr. Yi. He was a professor of Emory University. Now he's back in his hometown in China. Who published paper in Nature that FSH blockade improved cognition in mice with Alzheimer's disease. Did you see a difference in male versus female in stem cell levels maybe after
So,
a certain age? Yeah.
well, so the gender difference in Alzheimer's disease is well known. Females are at greater risk. The severity of their disease is usually greater, and the onset is earlier, generally speaking. Um, when it comes to neurogenesis, we have not seen a gender difference. That's not to say that there isn't. It just means that we have not detected one yet. But again, there are so many aspects to study that I would not dare to say that it doesn't exist. I would just say that we haven't detected one yet.
Yeah, thank you. It was just regarding the pre- precautions because we were discussing if women should just, take hormones to prevent the FSH contribution to Alzheimer's and so on. So, yeah, that's why I remembered that research. So, yeah, thank you. And Elizabeth, did you want to ask a question related to glia?
It's in the chat because I am thrilled to meet somebody who has a brilliant intuition in the subject. And so I am asking for your intuition. Oh, so the question is, what is the mechanism of action for slow auditory processing in dementia? Would that be gliosis? It doesn't seem to be possible that that would be related to hippocampal function.
That's a good question that I don't have a good answer for, unfortunately. It is. I think it's increasingly recognized that there is a deterioration of auditory function in Alzheimer's disease. What is the mechanism of this phenomenon is not known, and I don't know. So I cannot comment on
Can
this.
you,
I'm sorry. Yeah,
oh,
go
go
ahead.
ahead. I was just going to say, could it be possible that it's the relationship between the hippocampus and the prefrontal cortex that causes the deterioration of the auditory processing? The
Absolutely.
relationship.
Absolutely.
Yes.
I cannot rule it out. It's definitely a possibility. We see a lot of pathology in the prefrontal cortex, and you may be right. I think it's definitely worth pursuing.
Thank you. And did you really say that the female, you had mostly female mice in your study, right?
Correct.
And did you find that there was, that the female mice had worse or earlier degeneration?
They do. The mouse models of familial Alzheimer's disease clearly show an earlier and more severe phenotype for females, and their pathology progresses faster.
Does that depend upon whether they're housed with males or housed, does that does it depend on the housing? I mean, their interpersonal relationships,
It does.
inter-mice relationships?
So I would say that it's independent of the housing, though in our case we keep them separate from the males because when males and females are kept together in the same cages, this is considered a breeder cage, which changes the whole dynamics. And interferes with the whole. I mean, if the female gets pregnant and there's pups, it changes the whole dynamics in the cage and introduces additional factors that would be very difficult for us to control. And therefore, we always keep females and males when it comes to experimental groups separately. I don't know what the outcome of the pathology would be had a specific female had some pups and had to nurse the pups and so forth. I have no idea. But I could say that if you compare the same age group, the same genotype in mice, in female mice versus male mice, the pathology and the onset would be earlier, and the pathology would be greater in the females.
I had a follow-up question from the audience. How did you measure anxiety exactly?
There are several tests that we're performing. One is the light dark test. There's the elevated maze. There are several tests that we perform on a regular basis to assess anxiety.
I see. Are there any blood markers or anything like that also?
I'm sorry, what kind of markers?
Anything in blood work or?
Oh, I'm sure there are. We have not done that.
Got it. Thank you.
Sorry, can I ask one more question? I had the question. I mean, this guy, for me, it's kind of fascinating to hear his approach, but I don't want to focus it too much on one person. His philosophy is based on his clinical research, that his perspective is that the plaques in the brain are more a function of healing than a product which would be a key to treatment. And so he feels like I think in the treatment domain there are different drugs being tested. Some just enhance cognitive function, and some I think trying to go after the plaque. And I wonder if 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 you can follow his little unique, maybe a little bit at the rim of the medical spectrum perspective that it might be smarter not to go after the plaque, but to accept it as a sequelae of something going wrong and go after the going wrong first.
Yes, you're right. So the perspective of the field has been changing in that regard. Back in the days, we used to think that amyloid plaques are the to go after thing. Obviously, amyloid plaques are abnormal accumulations of whatever it is that sticky proteins that accumulate in those areas, and obviously they occupy space if nothing else, right? But they're not supposed to be there, and they're not part of the healthy brain, right? So we would agree that these are probably lesions, right? Whether these lesions are actually um, helping get rid of toxic um, peptides or other substances, that may be. Um, and the field has been moving towards hypothesizing that indeed these are the soluble parts that eventually get aggregated in the form of amyloid plaques. And therefore, it is not so much the amyloid plaques that we need to go after, but more of the soluble toxic peptides that may be the precursor of those plaques. And I think that's what you're referring to. Um, this is, this is definitely a possibility, and it is definitely, something to look into. And there are many labs that are looking into this possibility, and to go after the earlier precursors that have a putative toxic um, influence on different cell types. So this is definitely a possibility that I cannot exclude, and it's definitely valid. And I I think that it makes sense. And I think that again, like I said at the beginning, we have to re-examine everything as if we're absolutely wrong about everything, right? So I think it's good that people question what 100 and something years ago seemed to be like the ultimate hallmark, and it's actually not necessarily going to be the answer therapy wise.
Is it possible that those changes, those tangles, and are all healing from gliosis? And would you call gliosis a separate disease from from hippocampal damage, so that we could separate dementia and Alzheimer's and call them what they really are, the underlying processes?
I think there's a lot of evidence that neuroimmune processes take, major, have a major role in the pathology. It is very difficult to think of Alzheimer's disease without considering the role of the immune system and inflammatory processes. So I would say that it is very important to keep studying and understanding the role of immune cells and inflammatory um, substances in the pathology of Alzheimer's disease. I think it's again, as we have been discussing during the last hour and a half, this is a multifactorial disorder. There are a lot of things going on in simple words. So I think it is, the more we know about it, the more we learn, the more we understand these processes, the closer we would get to an effective cure. And again, it might be not, one cure fits all, right? It could be that people who have diabetes will have to have a certain intervention, and people who have, I don't know, a mutation, familial mutation, will have a different intervention. So it is possible that the molecular pathways along the way may be slightly different, with the same end result, the same cognitive phenotype at the end. And therefore, at this point, I would not exclude any of the above. I would definitely continue promoting research in all aspects and in all fronts, in order to enhance the possibility of leading to cure.
So the genetic would be called familial. The gliosis component would be called glial Alzheimer's disease. I'm just saying,
The,
if
the familial form is the genetic form. So these are mutations, autosomal dominant mutations in APP presenilins. The late onset, the sporadic form, is of an unknown cause, but there are a lot of risk factors. The gliosis is a part of the mechanism of the disease, generally speaking, whether it's a familial form or a sporadic form.
Yeah,
good,
thank you
thank
for
you.
answering those questions. I had one more comment or question about that you, in the end, presented that you could even see with in situ sequencing that the augmentation that you did to neurogenesis, that the profile was the profile of the engram cells was basically rescued also. Is that something you will pursue to find maybe more mechanisms that you can address, or are you looking into RNA sequencing? Like, what's the future or your ongoing research? Thank you.
Yeah, the hope is to understand the profile, the transcript profile of those new neurons, and try to come up with ways to correct, if you may, the transcript profile of the Alzheimer's new neurons. And this is definitely one of the approaches that we're taking moving forward.
If I, is there actually less likelihood or more likelihood in people with Alzheimer's to have epilepsy? I can't remember anymore. And because I know there is a form of epilepsy that if the stem cells don't incorporate well into the network, that that's kind of a severe form of epilepsy where then the neurons don't really integrate and just do, you know, random patterns and elicit epilepsy episodes. Um, is in people, does in people with Alzheimer's occur less epilepsy in their health history?
So epilepsy is an interesting aspect that occurs in a subset of the population of Alzheimer's patients. The full mechanism of epilepsy in Alzheimer's disease is not completely understood. There are more and more studies looking into this, and it is definitely possible that, and there are actually several very intriguing studies lately, published lately, that suggest a connection between hippocampal neurogenesis and epilepsy in Alzheimer's disease. And I think it's worth looking into very seriously. And I think that there's definitely a possibility that also, I believe it was Elizabeth who asked me about gliosis. So it's definitely possibility. There's a possibility that gliosis is involved in this process. But it's a very interesting phenomenon that we do not fully understand yet.
Sorry, I had one more question. Maybe I missed that in your presentation. The role of stem cell therapy at this point, I think it's probably experimental, and I'm not aware of any. Maybe there are some trials somewhere, but where is that status quo of that, and where that might go?
So, when you refer to stem cell therapy, are you referring to an intervention that modifies stem cells in the brain, or are you referring to stem cell transplantation?
The latter.
Okay. Well, it is definitely possibility that there's definitely a possibility that some groups pursue that. I personally, my group is relying on the fact that neurogenesis occurs naturally in the hippocampus, and therefore it would make it easier for researchers to find ways to modulate endogenous neurogenesis rather than transplant exogenous cells into the brain, if it makes sense to you. It's something that, if you think about it, especially as a neurosurgeon, you know that any intervention is partially an injury as well. So if we had to prefer whether we would like to modulate existing neural stem cells in the brain rather than injecting, stereotaxically injecting or peripherally injecting neural stem cells into the brain, we would prefer to modulate the endogenous
And
ones.
and what's the status quo of that former version to stimulate the brain cells yet already existing? Are there any studies or any controlled trials
So
available?
there aren't clinical trials available yet, but this is where we're heading. This is where my group is
I
heading.
see.
That's what we're hoping to achieve.
Yeah, I think that was a great last question to say that you're heading that way. And we've been going over 90 minutes, so I wanted to give you, um, a break from us. you Thank you so much for sharing your really amazing research with us, and I hope all the best for your research and future clinical trials. For me, when I think it looks really promising, and I'm very hopeful. So I really appreciate, and I think the feedback of these rooms and these talks are that people feel more hopeful about the future because of people like you and the work you do. So thank you so much. And yeah, I hope you enjoyed it and come back sometime. And yeah, thank you, Orly.
Well, thank you so much, and thank you for having me.
Yeah, and thank you everyone for coming and asking questions and commenting. We really appreciate it. That makes the discussion really interesting. And I hope I hear you all back soon. We'll have more rooms in the future. We'll have tomorrow Dr. Salis talking about how he uses how he uses biophysics and machine learning to design organisms. And then we'll have Dr. Venkat Arami talking about glioblastoma and how it manages to invade the brain. And on Friday, we'll have Dr. Goodyear talking about how he developed a technique to visualize heartbeats in vivo with an antibody dye. So yeah, thank you so much. Thank you, Orly, that you shared your precious time with us, and we wish you all the best. And thank you. Thanks, everyone.
Thank you.
Okay, I'll close the room in 3, 2, 1. Bye, everyone.
Bye.